Hello students, now we are going to discuss about the DNA ligase. That is the another enzyme which will be playing its function into the process of cloning. So let's start with the video. DNA ligase, in the sense DNA ligase, it catalyze or initiate, or it will it will catalyzing the particular formation of phosphodiester bond. Ultimately the formation of bond. During the cloning process, we are taking the DNA from the desired organism and we are inserting into the host cell, right? So there the DNA ligase, it is playing or it is initiating the formation of phosphodiester bond between the 5' phosphate of one strand of DNA, or it may be RNA, and the 3' hydroxyl of another, right? So that's what we have seen into the DNA structure, like 5 days it will be there, so then here C, T, A, something like that, then 3 days it will be there in another side, here 3 days, then again C, T, A or something coding, then 5 days, right? So whenever the another DNA it is going to attach with this this particular side, it may be the sticky end or it may be the blunt end or blunt cut, right? So there the particular bond it is going to form by the DNA ligase, right? So that is the mechanism that's very important into the cloning process, right? So this enzyme or DNA ligase is used to covalently link or ligate, that's what, bond formation or ligation of the fragments of DNA together, right? Because two DNA is participating into the cloning process. What is happening in cloning? We are taking DNA 1 and DNA 2. Those DNA has to be bonded together or linked together or ligate together, right? That's what, so that they can further develop for the development of the particular organism that is going on, right? So that's what it is covalently linked. Okay, so there are different types of DNA ligase which is there, which we need to understand. Like there are 4 types here. Basically I have written here: bacteriophage T4 DNA ligase, the second one is the E. coli DNA ligase, third one is the tag DNA ligase, and fourth one is T4 RNA ligase, right? So let's discuss bacteriophage T4 DNA ligase. It is nothing but it is monomeric polypeptide, right? Monomeric polypeptide, which is most frequently used in DNA ligase. That's what we are. It is derived from bacteriophage T4. That's what it is known as bacteriophage T4 DNA ligase. Second one is the E. coli DNA ligase. As the name indicates, E. coli is the name of the bacteria which is derived from E. coli cell, and it is a monomeric enzyme once again which catalyze bond formation in duplex DNA, right? That is the speciality of this particular ligase. Duplex DNA, it is going to form the phosphodiester bond, it is going to form. The third one is the tag DNA ligase. Here thermostable ligase, this is known as a thermostable ligase used in detection of mutation, right? So that's what this is the feature of this particular ligase. The fourth one is the T4 RNA ligase, right? As the name indicates, ribose nucleic acid here we are going to have. So whenever in cloning the RNA is involved, right? In RNA we are going to do some kind of cloning, then this kind of ligase it is going to be useful. It is the only phased RNA ligase. Yes, this enzyme catalyze or initiate bond formation in RNA molecule. That's what name itself is, T4 RNA ligase, right? So 3 it is the DNA ligase and 1 is the RNA ligase. So ligation is the one process where generally what is happening in when we are preparing any puncture, you might be seeing into the any garage, what they will do, wherever there will be the leakage, they'll put some kind of liquid and they are trying to fill that particular hole which has been created. So the same thing it is happening here. One DNA we are taking it from the one organism, desired organism, which is having best characteristic and feature, and with the host cell we are trying to ligate that one, right? That's what that DNA has to attach with the another DNA. So that this DNA ligase, this enzyme, it is going to useful, which is helping into the formation of phosphodiester bond between the 5 days and the 3 days, which will be there into the DNA structure, right? Thank you.